Welcome to part 4 of the Central Dogma series. This video will explore the construction of a protein and the process of translation. If you would like to learn about nucleic acids, DNA, DNA replication, RNA, and transcription, check out parts 1, 2, and 3 of this series. In the previous videos, we talked about the building blocks of genes, DNA and RNA, what they are made of, and how they are built. When it comes to replicating DNA, the first portion of Central Dogma, 4 enzymes are required. Helicase unzips the double-stranded DNA. Primase primes the beginning of the strand to signal to DNA polymerase to attach and start building the new strand. The DNA polymerase of the leading strand continuously moves from the 5' end to the 3' end. The lagging strand is created in shorter, interrupted portions called Okazaki fragments. There are spaces between the fragments that are incomplete. Ligase glues the fragments together to complete the lagging strand. During DNA replication, all enzymes are working simultaneously along the DNA strand. DNA replication differs between prokaryotes and eukaryotes. Prokaryotes have one point of origin to begin replication and divide into 2 new cells while the DNA is being duplicated. Eukaryotes, however, have multiple points of origin throughout the DNA strand. This is then followed by cell division or mitosis. For transcription of RNA, RNA polymerase and DNA are required. RNA polymerase attaches to, separates, copies, and reassembles DNA to create RNA. DNA is made of a template and a non-template strand. The RNA polymerase copies the template strand from the 3' end to the 5' end. In a eukaryotic cell, the new mRNA strand will have a 5' guanosine cap and a 3' poly A tail added. After this, the introns are spliced to form a mature mRNA. In prokaryotes, there is no nucleus, so the ribosomes can bind directly to the nascent mRNA, starting translation before transcription is complete. In eukaryotes, the mature mRNA must pass through the nuclear pores and be delivered to the ribosomes in the cytoplasm. We are going to focus on the last portion of Central Dogma, the translation of RNA into a protein within the ribosome. Ribosomes are located in the cytoplasm of the cell and perform protein synthesis. Ribosomes themselves are made of ribosomal RNA and proteins. How do ribosomes know what proteins to make? Ribosomes start and stop translation by reading codons. Codons are a sequence of 3 nucleotides that correspond to an amino acid chain. Amino acids eventually form proteins. When we are referring to codons, there are start and stop codons. The start codon of the mRNA strand is AUG. This codon codes for the amino acid methionine, which is always the first amino acid in eukaryotes. The ribosome will continue to build the amino acid chain by reading down the mRNA strand until it reaches a termination or stop codon. In this example, the stop codon is UAG. There are 3 possible stop codons that can terminate translation. There are UAG, UAA, or UGA. Charts like this can help us determine what amino acid is coded for with the 3 nucleotides. Another example is the circular reference guide to determine the amino acid. Let's go back to our ribosome. Ribosomes are made of ribosomal RNA and proteins. Their job is to produce proteins from mRNA. There are 2 parts to a ribosome, a small subunit and a large subunit. Within these subunits, there are 3 binding sites, E, P, and A. The amino acyl tRNA binding site is where tRNA enters. The peptidyl tRNA binding site is where the peptide bond is created and the protein is built. And lastly, the exit site is where the tRNA exits the ribosome. tRNA stands for transfer RNA. At the 3' end, the amino acid is connected to an acceptor stem. The opposite end loops around and is the 5' end. The tRNA structure also includes a T loop, D loop, variable loop, and lastly, at the bottom is the anti-codon loop. The anti-codon at the bottom of the tRNA corresponds to the amino acid at the top. For simplicity, we will look at the tRNA in this form. The important parts are the amino acid at the top and its corresponding anti-codon at the bottom. At the ribosome, the small subunit binds to the mature mRNA. This is the beginning of the initiation step. The tRNA with the anti-codon matching the mRNA will enter the ribosome, move to the P bonding site, and then the large subunit of the ribosome will bind. This is when elongation begins. The amino acid, methionine, is always the start codon. Now the next tRNA with the corresponding anti-codon will enter the A binding site. A peptide bond will form between the two amino acids, and the leading amino acid will disconnect from the tRNA. The mRNA will be read from the 5' end to the 3' end. The ribosome will continue to read the mRNA. When the leading tRNA gets to the exit binding site, it will exit the ribosome, and the next corresponding tRNA will enter the A binding site. The process will continue by forming a peptide bond between the two amino acids, and the ribosome will continue down the mRNA strand. The new tRNAs will continue to enter and build the protein. To complete the protein, elongation must stop and termination begins. A specific stop codon enters the A site. Stop codons do not have corresponding tRNA molecules, but will bind release factors which release the amino acids in the ribosome subunits. The chain of amino acids is released and leaves the area of the ribosome, and we are left with a complete and functional protein. These proteins could make vibrant purple or yellow flower colors, even the size and shapes of the flowers. The proteins can determine how big a leaf can grow, or if leaves develop natural holes like the Swiss cheese plant mentioned in the first video. Central dogma is an important process for all organisms. Without it, we would not function. Every step of central dogma ensures biochemical processes occur to make functional products to survive.